Good. Well, thank you for all for staying here to the end of these talks. Let me thank Bengt for the very kind introduction. And I want to say what a huge pleasure and privilege it is to be back in Budapest, and particularly to be at this incredibly exciting meeting. So I'm delighted to be invited. You've heard in a number of talks yesterday and today about the fact that peptides and proteins can form the amyloid state under some conditions, and that this can be deposited in tissues. And what I'd like to do today is to talk about what one could call the amyloid phenomenon, and particularly to explain its significance in medicine, primarily because it's associated with a whole range of diseases that are now becoming perhaps the most rapidly proliferating diseases in the modern world. They're incurable, and they have enormous importance. So I hope to start at the beginning, how we got into this, and then explain what we're hoping to do in the future. So the story started with protein folding that you've heard about. We have perhaps 100,000 different types of protein within us. They do everything in our bodies, and most of them have to fold up into a unique structure in order to function. And the instructions for folding are, of course, encoded in the unique amino acid sequences of each protein. They fold into helical structures like myoglobin, or this is an SH3 domain with beta strands forming beta structure, and this is an enzyme, actually, acyl phosphatase. So these fold to these exquisite structures with highly sophisticated functional processes, but also very beautifully designed surfaces. So that if you look at a cell, this is a bacterial cell, it could be any other type of cell, biomolecules are packed together extremely closely at 300 grams per liter, and if you're not impressed, try and make up a solution of that concentration. And their surfaces are such that they can remain independent or specific complexes within that highly evolved environment. But every type of protein within every cell, and higher order structures like the ribosome or ion channels and so forth, have to assemble correctly for living systems to function properly. And we got interested about 25 years ago in a question of what happens if proteins don't fold to those correct structures, but if you like, if they misfold in biology. And it turns out that protein misfolding is the origin of many diseases. Because they're so important, if proteins don't fold properly or don't remain correctly folded, then there'll be malfunctions in the cell, and a whole range of diseases from cancer to diabetes are now known to be very strongly connected with this misfolding phenomenon. And this gives a very simple picture of this. Our proteins are folding and unfolding all the time in cells, and there's a turnover mechanism, important, for example, for the cell cycle. But proteins can misfold, and if they misfold significantly, there are really three types of things that can happen. They can end up being produced, but actually being sent to the wrong place in the cell or the wrong place in the organism, and there are diseases associated with these trafficking problems. Other proteins that misfold are picked up by very sophisticated quality control mechanisms and targeted for degradation. And these are so-called loss-of-function diseases. The classic example is, I think, the most common genetic disease in the world, which is cystic fibrosis, where the misfolding of a membrane protein as a result of many different mutations ends up in lower concentration in people suffering from these diseases. They don't process ion transport in the lung properly, and they end up with the symptoms many of you will be familiar with. But there's another class of diseases where misfolding in the cell does what most of us who don't fold proteins properly in the test tube are very familiar with. They precipitate. And so you get a series of diseases associated with these precipitation of misfolded proteins. And it turns out that there are a series of very, very interesting diseases. And so misfolding can result in the aggregation, the clumping together, if you like, of otherwise soluble proteins. And some of these give these intractable amyloid deposits in cells and tissues. And we now know the number increases actually every year. There are at least 50 diseases associated with the amyloid phenomenon. The best known, and we've heard about, is Alzheimer's disease, but also the prion diseases, remarkably type 2 diabetes. It used to be called late onset diabetes. It now occurs regularly in children, even conditions associated with medical treatment, and Parkinson's disease, Huntington's disease, motor neuron disease, and so forth. And there's several interesting things about these diseases. The first is they're virtually unknown 100 years ago. Alzheimer's disease was first described in 1906 as a curiosity. The second thing is that each of them is associated with a particular protein or fragment of protein that forms these deposits. There are other proteins associated with them in many cases. And these can either be full-length proteins or fragments. They can be unstructured polypeptides or highly structured proteins. And so there's nothing in common between these that one can detect between the proteins that give rise to these diseases. So they're all amyloid deposits, and the best known disease, I say, is Alzheimer's disease. It's the most common form of adult dementia. Something like 40 million people suffer from Alzheimer's disease now. That number will increase to 130 million, it's predicted, in the next 30 years. And in fact, if you look at that whole set of misfolding diseases, you can estimate that in the not very distant future, perhaps 10% of the world's population will be suffering from one of these diseases. Type 2 diabetes, there are already 400 million people suffering from them, and they're incurable. And so this is a huge problem for the human race in the future. So in Alzheimer's disease, there's this A-beta peptide, that's one you've heard about before. It forms extracellular plaques in the brain. There's a protein called tau, which subsequent to this, it's thought, can aggregate within cells. And these give rise to toxicity that causes the death of neurons, the loss of memory, and cognitive skills that one is very familiar about with these symptoms. But this is one disease. This is Alzheimer's disease, but the symptoms can be extremely different. For example, if you look at this condition, if you put people on dialysis for kidney failure for 5 years, the dialysis is not completely effective. Not all proteins are filtered out. Their levels change in the blood. And every single person, effectively, who's been on this for 5 years will get deposits of amyloid, not in the brain, in this case in skeletal tissues and in joints, which cause enormous amounts of pain and discomfort. So these are amyloid deposits, but in a completely different environment. There's another, just to give you an example, another type of disease is so-called cardiac amyloidosis, where amyloid deposits occur in the heart. And it's been recognized recently that perhaps 25% of elderly people who suffer from heart failure, it's exacerbated by the fact that these deposits restrict the movement of muscle tissue in the heart. There are other diseases. Here's a systemic disease where these deposits can occur in the liver, kidney, spleen, and so forth. Again, sometimes in huge quantities, kilogram quantities in this case, and these are fatal diseases. Normally the organs rupture from the load of aggregated protein. This turns out to be very important for us because in the mid-1990s we were looking at the folding of lysozyme, not thought then to be the most exciting protein in biology, but a few people have mutants in the lysozyme gene, single point mutations which generate these depositions, and suddenly from being one of the less important proteins, perhaps the less exciting proteins in biological function, it became associated with these incredibly important diseases. And we've done a lot of work on this, but I'm not going to talk about it today. So the first question I want to say is, what are these deposits like when you get them in these diseases? Well, whichever disease you take, if you extract the deposits, whether it's from the brain or from the liver or from the joints or whatever, and look at them under the electron microscope, the aggregates contain large amounts of these fibrillar structures. They're long, thin, they can be microns in length, typically 10 nanometers in diameter, lots of beta sheet structure, and they look essentially the same whichever disease you look at, even though the place of deposition and the symptoms are completely different. And so we really became fascinated by this, and to say, what's the nature of the amyloid state? What are these fibrillar structures that are so different from the normal forms of proteins that have been studied for many years? Well, I perhaps should point out that almost every single thing that we've done in science has been done by accident. They're normally experiments which don't work, which we then look to find out why our predictions were wrong. I've already said we got into this whole field because we're looking at the folding of lysozyme, which then turned out in its misfolded state to be associated with one of these amyloid diseases. And our work on the structure of these fibrils came again from an accident. And we were looking at the folding of a small domain. I think it's the most common domain in the animal kingdom, the SH3 domain. It has nothing to do with any disease. We were looking at it because we wanted to understand folding. We took it to low pH, so it unfolded, and we're going to take it then back to normal pH, so it refolded, and we could follow the processes. But the postdoc in Archibald, a really brilliant postdoc, made a very expensive sample with lots of isotopes in here, took it to low pH, put it in an NMR spectrometer, and then took a long weekend. And I could work out later this came from the Thursday morning to the next Wednesday afternoon, because I could work out from the kinetics. And when he came back, his sample had turned into a gel, and we got no NMR signal from it. He was incredibly upset. But it turns out that we knew that these lysozyme variants, when they formed fibrils, actually also turned into gels. So we looked in a sort of mad experiment in the NMR tube under the electron microscope, and to our amazement, they were full of these fibrils. This is the first photograph we took in the mid-1990s. And they had all the characteristics of amyloid structure associated with the disease. So we went from a situation thinking that these structures only formed in this set of diseases, 50 proteins or whatever, to realizing that actually, at least in the lab, you could generate them from other proteins. And so we went on to look at this in much more detail, and we looked at what you might think is the least likely protein to form beta sheet-rich amyloid fibrils. It's myoglobin, the first protein to be determined by crystallography. This is John Kendrew looking very surprised at his success in this area. You can see the fold of the protein. This is the helical structure. And we put this, again, under denaturing conditions, left it for a bit, spun down the sample, took off the top solution phase, which looked like this, but in the bottom were these fibrils that again had all the characteristics of disease. So we realized that surprising proteins could form these structures. So people said, well, you've been lucky, there's some sequence motif that you're not smart enough to recognize that must be coding for this structure. And a very persistent graduate student, Marcus Vandrich, was very irritated by this. So he took homopolymers, these are polypeptides where all the residues are the same, and showed that if you found the right conditions, you could get homopolymers to form structures which, these pictures look exactly like the fibrils you can get from Alzheimer's disease, for example, but they're from polythreonine. And so this showed that the amyloid fold, if you like, the amyloid architecture, is not encoded in the amino acid sequence. So we knew that the native states were encoded exquisitely by the sequence. These forms of structures of proteins are not encoded in the sequence. They're a property, if you like, the polymeric nature of polypeptides. And so we realized, or we suggested that this is a generic sort of polymer structure of proteins. These are the proteins selected for function by biology. The side chains interact together and force the main chain to go into these wonderful structures. And this is a model we made. We now have structures for the amyloid fibrils. This is actually of SH3, where we can see they're sort of protofilaments that twist around each other to make these long fibrils. And they contain these beta strands. It's a double beta sheet, and they hydrogen bond together. And so we imagine that because the main chain which forms these hydrogen bonds is the same for all proteins, that if this is the dominant interaction, then of course all these structures will look the same. So there'll be the biological form encoded by the side chains, and this other, I sometimes call it the real form of proteins, which is, like many polymers, forms aggregated structures. We've now determined the atomic level residue by residue structures of various fibrils. This is a fragment of a transthyretin associated with one of these diseases. And you can see here, this is these core structure, so-called cross-beta structure. The fibril goes up in this direction. The strands are perpendicular to this. And what you can see here in this double sheet is this whole array of hydrogen bonds that holds this structure together between the main chain atoms, which I say are common to all different types of proteins. And the main chain dominates the structure. The side chains have to fit together in it as best they can. So we realized that there were these very strong, highly ordered structures with these very strong hydrogen bonds. And we measured the stability of these fibrils, and we were astonished to discover that this, if you like, alternative generic state, has similar characteristics for all proteins, varying depending on how the side chains pack in detail. It can be more stable with these arrays of hydrogen bonds than the native state, even under physiological conditions, particularly for small proteins. And so not only are our native proteins just one form of structure, but actually thermodynamically, even under physiological conditions, they're more unstable, they're metastable relative to these aggregates. And so there's an inherent tendency for many of our proteins to convert into this structure. And so the question is, why don't we turn solid immediately? And the answer is, fortunately, that this process is rather slow, and I'll say why that is. So we realized that native proteins had an intrinsic tendency to form these aggregated structures. And so the question then came, what's the biological consequences of forming these types of structures? Why should they be associated with disease, for example? And here, just to give you a sort of overview, you all know that if you look at the chemistry going on in biological systems, everything is controlled by enzymes, that make sure you've got the right molecule present at the right time in the right place. And even though thermodynamically most of these compounds would rather be something else, carbon dioxide and water, for example. And we thought that actually all these different possible conformational states of proteins, when you make a protein, it can clump into a disordered aggregate, it can get degraded, it can fold through intermediates perhaps into higher order folded structures, they can assemble into oligomers and fibers and so forth. But we argued that actually they could also convert into this amyloid fibril structures, of course going through some sort of intermediate species as they begin to assemble together. And our view was these are all controlled largely by kinetic processes, and biology is arranged that you don't generally get the amyloid fibril. There are a few structures which use the amyloid motif functionally. So why don't we want to get the structure? Well, first of all, it's clearly not functional. These are very ordered structures. They're actually weight for weight stronger than steel. You don't really want them in your heart. But even worse things happen, because we realized when we looked at these sort of early intermediates, these precursors, often called oligomers, you can see in the electron microscope, which form and then develop into fibrils. They're misfolded, they've got the wrong residues, if you like, for that protein on the sequence. They're very sticky. And we could show, for example, if you add them to various cell cultures, whether they're anything to do with disease or not, they'll penetrate cells and stick to various components or disrupt membranes. So amyloid formation doesn't just get rid of function, it doesn't just disrupt tissue and organs, it generates toxic species within the cells and the body. And so we put forward, this is a very simplistic overview, this sort of unified view of all these diseases with all their different symptoms. And so under normal circumstances, there are actually all sorts of what you can call housekeeping mechanisms. They scour our cells and pick up proteins which are misfolded or beginning to aggregate and target them for degradation. That's why we can continue in our functional states for decades. And it's the failure of these mechanisms that gives rise to pathology, either because you get loads of this stuff accumulating in your liver, for example, or because you get toxic species that can cause cell death, for example, in the brain. So this was our general view, and we thought, well, why are these diseases now occurring? And we realized if you look at the sort of quality control and the regulation mechanisms, that these are all, if you like, modern diseases. Alzheimer's disease is occurring because we live a long time. The prion diseases, because particularly in Britain, we were clever enough to feed young cows and old cows, which already had these aggregates present. If you don't eat if you don't run around to catch your food and get lots of exercise, you can become obese, which is a modern phenomenon again, which is a huge risk factor for diabetes. And in the past, of course, we didn't put people on dialysis. It's a very good thing, but it ends up with forming these aggregates. So we thought these were linked to subtle changes in protein concentration, sometimes with mutations in familial diseases, or with long life where these regulatory processes fall, or with diet, for example. And this could then result in the aggregation of these proteins and ultimately a whole loss of the control system which ensures that proteins remain in solution. And I wanted to show a little movie which illustrates this very simply. What we did was to take Drosophila. I'll show you what this means in a minute. We put in there the 42 residue peptide, the human peptide, expressed it in the CNS. It doesn't do anything functionally in these Drosophila and the fruit flies. But we chose strains where you can see amyloid deposition and neuronal damage. And we then made mutations in the sequence that we predicted from our test tube experiments, if you like, would make this peptide a bit more aggregation prone or a bit less aggregation prone. For example, put a stickier group in or a charge group. And then this is a series of these. These are the wild type 4042. What you do is you put these in a test tube, the different strains, you bang the test tubes on the bench, the flies drop to the bottom. If they're happy, they climb up again. You get graduate students to count them as they climb up. And what's great about this is, first of all, this is increasing aggregation propensity. When you do the experiment, you can plot the graph straight away. You don't need to do anything else. You can see this amazing correlation. But it's a remarkable correlation because this, if you like, is physical properties. The aggregation propensity is a physical property. This is physics. It obeys the laws of physics. And this is biology. This is 20,000 genes in Drosophila trying to control this process. And the aggregation propensity changes by a few percent, but it's enough to cause the more aggregation-prone protein to cause massive neuronal damage. But if you make the peptide a bit less aggregation-prone, it's indistinguishable from not having it there at all. And you see this amazing correlation. So this suggested we're onto the sort of right lines. And so in the rest of the talk, I want to talk really about how these amyloid structures form and what we can do to understand how we can control that. So can we determine the mechanism by which proteins go from a soluble state into these aggregates? Well, it's a very complex problem because we realized, and this is brilliant work by Thomas Knowles, who was then a graduate student, and 7 years later, from being a graduate student, he's a full professor in Cambridge. He's quite smart, actually. And he realized that you could start off with a soluble form of a protein, a misfolded protein, if you like. There's a primary step where little oligomers form, little nuclei form, And these can then, in these fibrillar structures, they can grow by adding molecules to the end. They can even dissociate and shrink by losing them. And this is the primary step, nucleation and growth. But we realized that there were other things that could happen. The fibrils, even though they're very strong, will sometimes fragment. That doubles the number of ends, and so you get twice as many aggregates, they grow twice as quickly. And we also realized that these aggregates could attract the monomeric form and give a more rapid process for nucleation. It's called secondary nucleation. And so it's a very complex process with different rate constants for these different steps, many of which going on in parallel. But we can see that in order to understand the mechanism, we'd have to identify the nature of these different molecular events. And this is the only equation I'm going to show, I think. Here you don't need to understand anything about it if you're very biologically inclined, but a really great breakthrough occurred in 2009, again by Thomas Knowles, who took these different steps in the assembly process and wrote down the differential equations explaining the rates in terms of rate constants. And so you get this set of coupled nonlinear differential equations that everybody said was completely insoluble because it's so complex. But he, remembering a maths course he'd taken in ETH in Zurich, actually found a way of getting an analytical solution to this equation. And what was really exciting about this is it meant that if you took experimental data, and here's one set of it, this is time, this is the low concentration of an aggregation-prone protein, which aggregates very slowly. As you increase the concentration, it aggregates faster. And he showed that if you actually obtained a set of data like this and fitted it globally to this solution to the equation, you could extract the individual rate constants for these different steps and therefore find out what the dominant processes are at different times in the aggregation process. This is now skipping about 5 years of learning how to do this, but I just wanted to show you these data, and you don't need to see anything in detail from this, but this is looking in the test tube at the aggregation of this A-beta-42 peptide associated with Alzheimer's disease. And with Sarah Linzer Lund, who was doing incredibly careful experiments, we got together with her, and we looked at the aggregation kinetics of A-beta-42 as a function of concentration. You can see beautiful data. There's a high concentration, there's a lower concentration. And we actually looked at different conditions, shaking, for example, which enhances fragmentation. And this then enabled us to fit these curves and to get the details of the key mechanistic steps which dominate these aggregation processes. And we were very surprised. We thought these primary nucleation step, where you have to get the molecules to come together, were going to be the really rate-determining step. But it turns out it's not so. For A-beta-42, it's actually this secondary process. Once you've got aggregates formed, they catalyze the formation of more oligomers, which then more nuclei, which then grow to more fibrils. So the more of these you get, the more new ones you generate. And so this is a really explosive process. You know, it's once you start to get aggregation, it proliferates, the aggregates proliferate very rapidly. And so this turned out to be an incredibly important mechanism for A-beta-42, and we have strong evidence this is the case in vivo as well. So it raises the question, if when you start to get aggregation, it starts to get exponentially out of control, how does biology manage it? And so how are living systems protected against these processes? Well, I think you know that if you look at protein folding in the cell, there are molecules, particularly molecular chaperones, that associate with the early formation of these polypeptide chains, which are themselves aggregation prone, and they stop them aggregating until the proteins are properly folded. So they're called chaperones because they look after these non-adult proteins until they're able to look after themselves in this complex environment of the cell. But it turns out that these chaperones are not only important in the initial folding, it turns out, and again we're surprised by this, that they're very effective at suppressing aggregation reactions. And here's just one example of this. Again, this is work with Sarah Linzer and others in Sweden. We took a chaperone, it's called Bricos, it doesn't matter what it is, And if you add it, this is the aggregation, the absence of the chaperone. This is adding more and more chaperone. You can see it slows down the rate of aggregation. And then we took our analytical solution and said, well, what's the mechanism of slowing down? If it had been preventing the primary nucleation, it would just have delayed the whole process, which would then have gone off. If it was elongation, you get a different type of curve. And it turned out that this set of data fitted perfectly to a fit when secondary nucleation, this dominant one in the free A-beta-42 is important. This suppresses it. And it's very important because if you were, for example, to stop elongation, you get an accumulation of these precursor proteins, which would be horribly toxic. But by inhibiting the secondary nucleation, stopping these nuclei forming, you reduce dramatically the number of oligomers. And indeed, we know that this chaperone, if you put it in flies, for example, absolutely suppresses the damaging effects. We know how it happens. We can do immunogold labeling. We can put gold clusters along the surface, or we can do binding studies. It's really amazing. It's very simple. It simply binds, this chaperone binds onto the fibrils and therefore covers up the nucleation sites. And it turns out that we've looked at lots of chaperones, and biology is absolutely wonderful. It has hundreds of chaperones, and some of them inhibit primary nucleation, some of them inhibit elongation, some of them inhibit primary nucleation. So there's a whole army of these defense mechanisms that start at the beginning of the process, and then if that fails, and another defense mechanism comes up, and that's how this process is controlled. So in the last couple of minutes, what I want to do is to say that's how biology controls this process. But of course, as we live longer, these mechanisms start to fail. So can we find some way of enhancing these protective mechanisms so that they'll last for longer, if you like, or we'll help them by giving some additional protective mechanisms? And so we thought, let's try some rational arguments on this. And again, I just want to give you a flavor of this. We use the same kinetic analysis we used on the chaperone to look for other compounds. And this just shows a couple. Again, we were astonished by this. These are small molecules. I'll mention a couple of them later. And adding the small molecules, even at relatively low concentration, has a dramatic effect on the aggregation. If you choose the right small molecules, many have no effect. And you can get these huge effects by adding small drug-like molecules, in the test tube at least, that inhibit one or more of these steps, different steps on the aggregation process. Again, you don't need to see the detail. On the next slide, I just wanted to say we've now worked this up into a very high throughput screening strategy. We generate a library of potential small molecules that we think are associated with A-beta. We select some of those. Some are already drugs, some are new molecules. We look in experiments to see if these will inhibit the aggregation process. If they do, we look to see which step they inhibit. And so we get molecules which are potential inhibitors from this screening process. But this is in test tube. Does it work in living systems? Well, we are moving towards humans, but first we're looking at ones we can screen more easily. These are the C. elegans system. There's the nematode worm, which was mentioned actually in relation to plants. And we've actually set up an automatic system to follow the movements of these nematode worms. We used to be able to do 500 worms a week. Now we can do 1 million worms a week by doing this in parallel by a tracker device. And I just wanted to show you a couple of the results of this. This is the C. elegans, which is transgenic for the A-beta-42. In other words, the A-beta-42 is being expressed in the muscle of the worm, and it causes paralysis. These are very, very unhappy worms. They're not doing much apart from sort of jiggling around. But if we add a small molecule, this actually is a molecule called bexarotene. It's actually already in use as an anti-cancer drug. And if we add that to the medium which the worms are developing, it has a really dramatic effect. And we have some very happy worms here that are moving around. This is all they do when they're happy. They just jiggle around like this. So it's fantastic news for demented worms. You know, we can treat them completely. But I think it's dramatic, and we've now actually got molecules that we've developed which are 1,000 times more effective than this already drug. It's an anti-cancer drug. You wouldn't want to take it for long periods of time. And so we think that there are many, many opportunities to begin to think of intervening in this process of amyloid formation to help, if you like, on natural defense mechanisms. And you know, we've done experiments. We can find some small molecules that inhibit the primary steps. We can find some that inhibit the secondary steps. We can even find ones that inhibit the growth and potentially even fragmentation by binding to the surface. And so we think that it might be possible to find small molecules which, if they work in people as well as worms, would actually be able to inhibit primary nucleation. If you could do this, it would be like a neurostatin. If you could stop primary nucleation, you don't have to worry about anything that happens later and what happens in biology. It would be a protective molecule. If you're my age, you forget about this and you start thinking about the proliferation step. But if we can find molecules that inhibit this process, then we should be able to stop the proliferation of the aggregates, and that may be able to control the spread of pathology through the brain. The nice thing about this is it doesn't just apply to one of these amyloid diseases. We've looked, for example, at worms which express alpha-synuclein associated with Parkinson's disease. We found other molecules. This is a natural product called squalene. And we have many other molecules now, and if you put this in worms, they're extremely happy, and you've suppressed effectively the toxicity that generates from the aggregation of the A-beta, in this case alpha-synuclein. So it seems if this method works, it could be generally applied. And what we're doing now, well, this has really happened in the last 5 years or so, is we've now raised about $50 million, and we're putting a building up. And this building is exciting. We call it Cambridge Centre of Misfolding Diseases, because it brings all of us who are together in one building associated with the chemistry department to do the fundamental science. And actually we also have space for a new drug discovery compound, which is taking our scientific, if you like, compounds in these simple systems and taking them through to, we hope, ultimately the clinic. So I wanted really to end up by saying, you know, Alzheimer's disease is a terrible disease. All of you probably know somebody, relatives of friends who've suffered from this. You can't really do anything to control it. It's 50% of you will probably get Alzheimer's disease if we don't succeed. If you live to 85, you've got a 50% chance of developing the disease. Just to cheer you up, but to cheer you up more, I'm very optimistic that actually science will triumph, as it has with all diseases in the past. And so this is actually Alzheimer's disease in the US. 2015, the US spent $200 billion treating people. So not treating, caring for people with Alzheimer's disease. And it scales by population. That's a lot of money. In 2050, it will spend $1 trillion a year just caring for people. There are now 5.5 million, there'll be 20 million. It's going up like this because aging population. It's not just, you have 1 in 100 chance at 65, you have, say, 1 in 2 chance at 85, and the number of people over 65 is increasing. In fact, it's now stated or thought that of all the people in the history of humanity who've reached the age of 65, more than half are alive now. And so that's why it's becoming such a massive problem. But we think, you know, by following up accidents, bringing together lots of techniques, we might be able to understand how this aggregation occurs, how biology controls it, and then if we're lucky, we can use that information to act as a basis to develop selective strategies. And I did nearly all the experiments myself, but there are a few other people involved. We have a whole lot of fantastic international students, this is just some of them. But what's been really fantastic for us is we have a group of people in Cambridge, there are really 4 of us, who share our students, funding, space, and so on. We work very closely with Sarah Lindsay, and we have lots of other collaborations. that have many long-term to help us do the experiments that we couldn't possibly do. So I hope that I've given some idea of what we're doing. I hope I've pointed out that you better get involved in this lot if you don't want to either spend all your money in the future looking after people of my age or suffering the same fate yourself. But I think the good news is science is developed. It's a golden age of science. Its techniques are now becoming available. And I hope that lots of you get involved in science and tackle issues like this, which otherwise, are really going to be a major issue in the next 20, 30 years for people, not just in rich countries. 70% of patients in 30 years' time would be in low or middle income countries. So thank you very much for your attention.